You do know if you yell at me or insult me, I got proof, right? I can go back and hold that against you. They said this about me, Your Honor. Okay, so let's talk about chapter 17. Okay, so again, this is a review. It's 50 minutes about, so I can't take forever on it. So, but if anyone, and again, my rule is this: if you got a question, shut me up, and we'll talk about it. If I'm going too fast, say, hold your horses. Let's go back a step, slide. I don't mind. That's great. Everyone writes and learns differently, so if it's a problem, just yell out. So, what is endocrinology? I would expect you to know what endocrinology is. Okay? It's just the science of hormones. Or the science and the study of hormones. You should know what a hormone is. Right? At the very base level, what is a hormone? It's some type of a chemical signaling molecule. And we talked about lots of different types of chemical signalers, right? We talked about ones that flow through the blood, some that go very short distances, some that loop back and affect their cells. But ultimately, all of them, at the end of the day, are going to be chemicals that are moving around. A chemical signaling molecule. And the other thing here is, and you can appreciate this fact after we went through this, is what? Hormones tend to be made in one location, and they move to other locations to have an effect. And you can appreciate that fact by looking at, for example, the pituitary gland, right? Definitely appreciate that. Okay. You should also know this, and kind of going back to that kind of general paradigm. This is for circulatory hormones, right? We have a secreting cell, blood vessels, and a target cell, right? So the core here is what? Some cell that makes the hormone. Typically, a blood vessel carries it some distance. Either that can be a very short distance or a very long distance. And then it goes to some type of a target cell. And on the target cell, we either have a receptor on the outside or on the inside. And that's just going to depend on the hormone. If you're talking about insulin or glucagon, we're talking about a receptor on the outside. If you're talking about T3 or T4, you're talking about a receptor on the inside. You're talking about testosterone, estrogen. You're talking about receptors on the inside. The secretory cells, these are cells that make the hormone. Think of the anterior pituitary. Right? Those hormones are made in that area. Those are secretory cells, or secretory body, glands. And then this distance right here can definitely vary. Sometimes it goes from the base of the skull all the way down to your gonads, for example. Other times it'll go from the base of your skull to your thymus. Just depends. It just depends on the situation. Okay. I would expect you to know some of the differences between the endocrine system and the nervous system. What's the key point where they're the same? What are we doing with both? Exactly. We're communicating information. With the nervous system, we're communicating information from, say, our sensors on our hands to our brain, or from our brain to our muscles. The endocrine system also does communication. But you have to know some of those differences. Right? And I go back to this analogy here, it's very much like the game Operation versus Mousetrap. Operation, you touch the side and you get an instant buzz. Well, instantaneous feedback, right? That's the nervous system. Mousetrap, on the other hand, you start the ball going and the cage goes down and, levers go up and down and balls go all around. That's the other part of it. That's more like the endocrine system. It's this multi-step reaction. And you kind of appreciate that. Remember how glucagon worked? Right? It set on the outside and then all these things happened on the inside. You can definitely appreciate that analogy. So responsiveness to starting, nervous system is fast, endocrine system is slower. Stopping is the same thing. Nervous system stops real fast, endocrine system starts really fast. Before I continue, let me kind of give you an idea of what kind of test question would come from this. I could say, in a short answer, name 3 differences between the endocrine system or the nervous system. Or I could say, true or false, the nervous system stops sooner than the endocrine system.
Are you going to be short answer on the test?
Short answer as in a sentence, perhaps, or 2 sentences, not write me an entire book. Not like the first test where it's this long essay that I have to go and correct. It would be more like, and the longest thing would be write 3 differences here, and you'd have to make like a 5-word sentence. Okay, not fill the entire page up. Okay. Should read those. You won't know that. Glands, I want you to know what a gland is. A gland is an endocrine or hormone-secreting body. Good, true, false question. Are glands the only type of hormone-secreting bodies? False. No. Organs do it. Hearts do it, right? They release a peptide. Other things have endocrine-like functions. So don't think that glands are the only thing that release hormones. Bless you.
The glands are hormone-secreting
Bodies. But don't take that to mean they're the only hormone-secreting bodies. Lots of our organs, for example, the heart, can also secrete hormones. So they're not the only game in town. Okay, we talked about a lot of the endocrine glands, and again, these are not the only things that secrete hormones. But we talked about a lot of these in class, right? We talked a lot about the pituitary. Talked about the thyroid with T3 and T4, mostly T4. Talked about the parathyroid hormone. The adrenal glands, pineal glands. We talked all about these in differing amounts. Okay. Here are some glands, again, that do not have an actual gland function with the endocrine system, but they do release their own hormones. I'm not going to hold you to know all of those. Just have the idea that, just because it doesn't say it's a gland does not mean it does not release hormone. Okay. An incredibly important thing here, this is really important, are these pathways of hormones. Okay, one type of hormone, and this is the more common, is this idea of a circulatory hormone, right? Hormones are made by some endocrine cell, say the pituitary. They travel through the bloodstream and then to their target cells. And most of the hormones we talked about fell in that category, didn't they? Insulin, made in the pancreas, goes throughout the body. Luteinizing hormone, oxytocin. Okay, a lot of hormones we talked about would group under these circulatory hormones. We talked about a few of these paracrine and autocrine hormones too. You should know what a circulatory hormone is. Circulatory hormone is made in location X. It goes along the circulatory system as a conducting pathway and winds up with some target cells at the end. The distance here varies, right? How this hormone moves through varies. Sometimes this hormone moves as a separate entity, right? But other times, what do we have with these hormones? We have with the insoluble ones. They are what? Sometimes a carrier protein that will move it along. Remember the target cells. The receptors can be on the outside here, but they can also be on the inside. So just don't see the picture and say it's just a receptor outside. They can also be on the inside. Okay. Local hormones, paracrine and autocrine. I would expect you to know what a paracrine hormone is and a autocrine hormone is. Paracrine affect nearby cells. Autocrine affect the same cell. Okay, so if I said, and you buy, that would be a good short answer, not short answer, but a fill in the blank. Blank hormone is a hormone that's released by a cell and affects the same cell. That's a autocrine. Exactly. Okay, we gave some really brief examples of these. This is one that you will see coming up in the immune part, and we talk about how T cells are activated. When we talk about that, we're really talking about a paracrine type pathway. Now, I'm not going to hold you to know that specific example. I just use that to show you the type of distances we're talking about. Another one, and this is probably one that you remembered, nitric oxide and the activation of a erection. But that is a paracrine type hormone. Okay. Another important concept, hormones are not multi-purpose things, right? They only have a work against their receptors. So insulin will not affect a glucagon receptor. Glucagon will not affect an insulin receptor. They are specific for their receptors. They're not multitaskers. There's no general purpose hormone out there. Okay, kind of the same idea. And before I continue, I want you to get the idea. How do hormones work? Tell me how they work. They're going to bind to what? Receptors. And where can these receptors be located? On the outside from the inside, right. The outside, going back to all that minutia, that stuff that's on the board with glucagon, that's one of those outside receptors. Okay? That starts something on the inside of the cell that leads to a bunch of changes on the inside. We saw all of that before, right? Other times, T3 is a good example of this. T3, it enters into the cell and goes to a receptor literally inside the nucleus and turns on the transcription of mRNA and then the translation of mRNA. So we're actually physically turning on genes at the molecular level of these hormones. Target cell. Target cells have about 2,000 to 100,000 receptors. A lot of these pictures you're going to see in your book and on the internet, you're going to see a little round ball and maybe 10 or 15 little receptors on the outside. We don't have the ability to show you the hundreds of thousands of receptors on the outside or the thousands of receptors. There's a lot of receptors. Okay. The book, the pictures you see are just there to give you an idea of it, but don't think that every cell has 10 receptors for hormones. Okay, it has tens of thousands. It's counted in the tens of thousands of receptors. And not only that, the numbers of receptors, is the number of receptors a variable or is it a static thing? It can vary. Cells can turn on the production of receptors. And if we have more receptors present, what does that tell you about how sensitive that cell is to the hormone? More receptors, the more sensitive it is to that hormone, because more of those things are coming in. Going back to how glucagon worked, right? If there's more receptors for glucagon, what's going to happen? It's going to make more of that cyclic AMP, right? It's going to turn that system on a lot faster. Okay. The idea of up and down regulation, I would expect you to know what, if I say if something's down regulated or up regulated. So if something reduces the number of receptors, that would be a down regulation. Up regulation, you have more receptors. Believe it or not, people actually monitor the mRNA that codes for these receptors, and they'll actually use that to study cells. I won't go into the specifics, but I mentioned this when I talked about it last time, that I knew someone at UAB that's actually doing a PhD right now doing that. Okay, so know what downregulation is and upregulation. But the point is, if it's upregulated, it's more sensitive to a hormone. If it's downregulated, it's less sensitive to that hormone. Types of hormones, you have to know the different types of hormones. Okay. You have to know the different types of hormones. What are the two major types? If I could break them down into two groups of hormones, what would they be? Chemically wise. Water soluble and lipid soluble. Okay. The water solubles, I probably would not, as I said, I would not ask you to draw this out on a test. But I would expect you at least to know what's going on, right? Water soluble, are those going to enter into a cell or stay on the outside? I'm sorry? The water solubles are going to are they going to enter in or do they stay on the outside? They stay on the outside. So remember this general thing. Again, for the test, I'm not going to hold you to know what PDE does, what cyclic AMP does, what cyclase does. We're not to that point. But I want you to see that cascading reaction going on inside. Right? It's that mousetrap idea. Something sits on the outside, causes some change on the inside. That leads to a change. 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 And ultimately something happens in the cell. And a great example of that is the idea of glucagon. Right? Another idea is with the idea of insulin. Same idea. One thing on the outside, a mess of changes on the inside. Other ones were the lipid solubles. T3, T4, we can group those in here. Thyroid hormones, your steroid hormones, your estrogens, your testosterones. What are these going to do? Go on the inside or the outside? Inside the cell. And because they're not soluble in water, what do they have to go with? A transport protein, a carrier protein. Because blood is liquid, right? So it has to be carried with a transport protein. Correct. And then it stays on the outside, and then when it interfaces with what on the outside? A receptor. It causes some type of internal change on the inside. And not just a small internal change. It's a multi-step change that leads to many things. Okay. The lipid solubles, these are going to do what? Go on the inside of the cell. Right? They're going literally to the molecular level. And where's the receptors for these proteins, for these hormones? Where's the receptor for these hormones? Inside the cell, exactly. They have to, just because a hormone goes in there, if that receptor's not present, it shows off. It's not going to do anything. It's not going to do jack. It has to have that receptor on the inside. Okay. And ultimately to the production in this. Talk about the pituitary. And again, what do glands do? They release hormone, okay? We should know what the pituitary has two parts, the anterior and posterior. For the test, we're not going to worry about these nucleuses and all that stuff. We're not going to worry about that. But what we do want to know is what? What's the connection with the pituitary and the hypothalamus? Exactly. The hypothalamus will control the pituitary. Now, in some cases, with the posterior pituitary, the hormones are made here and taken to the posterior pituitary. In other cases, there's these inhibiting releasing hormones that go to here, they travel to the anterior pituitary and tell the anterior pituitary to either release more hormone or stop releasing hormone. Okay. So for the test, what would I expect you to know? I would expect you to know what's closely associated with the pituitary, and that's the hypothalamus. And then I would also expect you to know that some hormones are made in the hypothalamus and taken to the pituitary to be released, and in other cases, hormones are made in the anterior pituitary, but their release is regulated by the hypothalamus. A better picture of this, I guess if you want to use this as a way of thinking about it, these bona fide neurons the hormones are made here, and they go physically to the posterior pituitary where they're released. Oxytocin is a good example of one of those. They release the hormone here, and it goes and travels to the anterior pituitary and controls the anterior pituitary from there. Okay. Let's not worry about the intermediate lobe. The intermediate lobe goes away after birth. It's not an important point here. I don't know why I put it in there last semester, but I did. But let's not worry about that one. Okay. For this one, we're not going to get too specific into it. Just know the pathway. And we talked about this before. I sound like a broken record. Hormone is made. Hormone is made here. It enters into this plexus. I'm not going to explain what a plexus is, but it enters into this capillary bed. And the hormone, then the releasing or inhibiting hormones travel to the anterior pituitary and release into here and control the pituitary. So that's kind of the point of all those. We're not going to get into the super specifics of primary and secondary plexuses and things like that. But I want to give you the idea that the hypothalamus is controlling the show here. That's the key point. No, you mean that what inhibiting and releasing hormone is? Yeah. We're not going to get that specific. But I do want you to know that the hypothalamus is releasing hormones that turn on and off the pituitary.
Now, the anterior and posterior, the day we talked about it, was it the anterior that made hormones and that were controlled by the hypothalamus?
Correct. And the posterior pituitary have the hormones made in the hypothalamus and go to the posterior pituitary for release. Right. And to make that point, there's a picture in here that really does hit that point right here. Posterior pituitary is made here, and they travel. They travel to the posterior pituitary. Here they're made here, but those releasing inhibiting hormones go release through here, and then go to the bloodstream, the capillaries, and into the anterior pituitary. But the hormones and the anterior pituitary are made in the anterior pituitary. Okay, I want to just get to the specific hormones here. So we're going to start talking about the pituitary hormones. First pituitary hormone is growth hormone. Growth hormone does not affect, well, first of all, growth hormone directly does not affect the tissue. Actually, it's going to stimulate an IGF, an insulin-like growth factor, but ultimately, the actual end result of growth hormone is tissue growth. Next thing with growth hormone is it affects tissue and bone, but it affects them differently. Right? If you have an excess amount of growth hormone, you're typically going to grow taller when you're young. But what happens in your late adolescence? What with your bones happen? At AMP1, what happens with that plate? What's the plate I'm thinking about? Epiphyseal plate hardens, we don't have bone lengthening. But growth hormone, it's not going to make that bone longer, but it's going to make them thicker. It's going to make tissue thicker and grow, but your bones are not growing longer. So what does that mean? You get things like acromegaly. You get flat bones, like facial bones, those are growing, right? But the bones in your arms are not growing, right? The flat bones, the cuboidal bones in your hand, those are growing, but your arm bones are not. Long story short, your hands become very knotty. Your face becomes very distorted because you have asynchronous tissue growth. Okay, some bones are growing, some bones are not. So what I want you to know about growth hormone, at the very least, it stimulates growth. It's going to work on bones and tissue, and then it works on these bones and tissue differently, right? Some bones, long bones, it will not grow, make grow longer in late adolescence. Other tissue, it will continue to grow throughout your life. Now, I didn't put the picture in here, but if you do search this, you will see actually people with this, and over time, their faces become much more just distorted. So when they're in their 40s or 50s, they're almost like caricatures. Okay. This one? Well, we talked, these were some of the broad groups, but we talked about each one of these groups specifically. So, if anything, you'd have to know it when we talked about those hormones specifically. So these were just broad groups where I kind of introduced them, but what I would test more on, or what I would test on, is like testing when we talked about T3 and T4, when we specifically talked about T3 and T4. These are just some larger groups of them, like somatotrophins. I wouldn't really ask anything about somatotrophins, but I would talk about growth hormone. And this material I would pull from that would be from when I talked about growth hormone here. Okay. Thyroid stimulating hormone, what I want you to know about this one, you have to know about how goiters develop. You have to know that. So how does a goiter develop? Lack of iodine. Why is that important? Tell me paint me the picture. So let's start this show from the hypothalamus and the pituitary. The pituitary releases what? What does pituitary release? It's up there. Big black letters. TSH, thyroid stimulating hormone. Okay, great. So the thyroid, the pituitary releases thyroid stimulating hormone. And what's the target cell? What's the target gland thyroid stimulating hormone? The thyroid. All right. And then when this thing interfaces with the thyroid, what's it going to do to the thyroid? Well, look at the name. It's going to stimulate the thyroid, exactly. So, hold on, guys. Thyroid stimulating hormone stimulates the thyroid to make more thyroid hormone. Okay. That's the first part you have to know. What does the thyroid need? Let me say this way. What hormones does the thyroid make that work? T4, more so T4. Let's just say T3, T4. But more so T4. What does T3 and T4 need? What do those numbers refer to? Iodine, exactly. So to make functional thyroid hormone, we definitely need some iodine here. Okay? When it functions correctly, you release the T4, it becomes T3, it becomes active, and it goes through the body. But what happens when it does not? What happens when you do not have iodine? What happens? Exactly. Your hypothalamus detects that lack of good thyroid hormone, and it doesn't understand it's from lack of iodine. It just says it's not being produced. So what does the hypothalamus and the pituitary do? Exactly. The hypothalamus says to the anterior pituitary, make more TSH, make more TSH. So it makes more TSH. And what happens to the thyroid? It is stimulated more. It's overstimulated to the point where what happens? A goiter forms, exactly. So goiter is from the overstimulation of the thyroid hormone. Because, in the majority of cases, you do not have iodine to make the active thyroid hormone. Lack of active iodine, you don't make this. Your hypothalamus detects as a lack of it, tells your anterior pituitary to release more thyroid hormone. It overstimulates the thyroid, and there you have it. And we really don't see that very much anymore, and because we have iodine in our salt. But I would expect you to know about this. And the reason I want you to know about this is because if you're a nutrition, you should know about it with that because of iodine deficiency. And some people have this idea of using just sea salt on their food, and it's not what I do. But and then also, if you are a nurse that goes overseas, people have a goiter, you should have an understanding on a physiological level of what's generating those goiters. FSH and LH, follicle stimulating hormone or luteinizing hormone. We're going to save this discussion for about a few weeks from now when we talk about the reproductive system. But for right now, just know these are involved in reproduction. Okay, FSH and LH. These are released by the anterior pituitary, and these are going to target our gonads. For this test, that's what we're going to leave it, right? We're going to get into the specifics of how these work when we talk about the reproductive system. You're going to see these in the future, but for now, just know that they have a function. Their target is our gonads. Prolactin, milk production. Don't confuse this with milk letdown. Adrenocorticotropic hormone, ACTH. Target here is the adrenals. MSH. If you have way too much of this, you can actually stimulate melanin production, but that's an overstimulation of it. Different effects in different organisms. It does not have let me say it this way. In humans, MSH does not have a tremendous effect as it would in other organisms. Okay. In other organisms, melanocytes, that's a lot more important in physiology. For humans, not super important, and it's not like the other hormones in that regard. So, okay. Posterior pituitary. The two hormones here are going to be ADH and oxytocin. ADH, I would expect you to know how ADH works, right? Remember we talked about that on the board. What does ADH? Here's a mnemonic if you want to help you out. ADH, think of always digging holes. So when ADH is released, what does that do to your ability to reabsorb water? Does it increase or decrease? So you're going to make it okay. Increase or decrease? Increases your ability to bring in water. Right? Think of it as always digging holes, making holes in that collecting duct in your nephron to bring in filtrated through urine. Probably important you know that drinking alcohol inhibits ADH, meaning that what you're going to usually do when you drink alcohol, you pee more because that hormone that tells your kidneys to reabsorb fluid, that's not getting what it should. That hormone is being blocked from release. That causes you don't reabsorb water as much, and the water is going to go back inside you or out. And in this case, it goes more out. Okay. No, the heart, the hormone from the heart. That's not one we're going to worry about. But someone had asked about a synergistic effect, and that's what I was addressing. Okay. Now, I'm not going to hold you to know what these aquaporons are, but what I do want you to know is what the release of this hormone causes the release of what? Or what does vasopressin or ADH cause? Basically. Very simple term. When ADH is present, what happens? Exactly. It is allowing, when ADH is present, these channels for fluid, it fuses with this membrane here, and it makes a channel for water to enter in. Okay. Going back, let me go back a picture here. Those channels are here. Okay, those pictures I showed you, those channels for bringing in fluid, is on the collecting duct. Okay, so when ADH is present, fluid comes through here, goes through the convoluted tubules, comes down here, and that the ADH is present. What's going to happen here? What the channels that allow water to come in, those are going to open up in effect. Those channels open up, water comes in, and does that mean more water comes out here or less? Less, exactly. You're reabsorbing water. Now that opening up was kind of a short way of saying it. Technically, it's a lot more complex than that. It's quote-unquote opening up. Instead of them opening up, what's happening is the vesicles that have those, they fuse with the membrane and allow the inflow of water. I'm not going to hold you to that level of detail, but I do want you to see that as these things open up, as these channels open up, the availability of water flow channels increases. Okay, does that work? I'm not going to hold you to the specifics about membrane fusing and everything else. I just want you to see that when ADH is present, the pathways for fluid reentry increase. Because it increases, we have more of a reabsorption of fluid. So we're good with ADH, because we talked a lot about that one. That's a really important one. And before we put past this, are we good with ADH? Okay. Anyone that says no? Okay. Talked about that one a lot. And let me test it a lot. Okay, oxytocin, don't confuse this with what? Prolactin. Prolactin is milk production. Oxytocin is milk letdown, the release of milk. Okay. Thyroid gland, you should know the two regions of the thyroid gland, the follicular and the parafollicular. The follicular are the larger cells here. The parafollicular are the smaller cells in between. Okay. What happens in the follicular cells? What happens in that? Before you get off the track here, what gland are we talking about? The thyroid hormone, exactly. Thyroid hormone. What does the thyroid hormone make? Major ones are what? T4 and T3. And I'm going to get too much into it, but mostly it's going to be making T4, and the active form is T3. So the majority here is going to be T4. But these are hormones that go inside the cell. They regulate regulate metabolism. Okay. If someone has too much of this, what do they have? If someone has too much of the thyroid hormone, what do they usually have? Hyperthyroidism. Okay, if someone has hyperthyroidism, what are they usually? They pencil thin, or are they really fat? They're hyperthyroidism. Thin, fat, which one? Thin, exactly. They are overstimulated their metabolic rate. If someone has hypo, H-Y-P-O, hypothyroidism, they're heavier, exactly. They're not making these metabolic hormones. Okay. Parafollicular, those are those little cells kind of between the follicular ones. This is going to make calcitonin. Calcitonin, what does calcitonin do? What does it do to calcium levels? Brings them down. What's the analogous. What's the antagonistic one to this one? Parathyroid hormone, exactly. Parathyroid hormone, this is going to bring calcium levels up. It's going to stimulate things that break down bone, for example. That releases calcium. You should have an idea of the calcitonin parathyroid cycle, parathyroid hormone cycle, I should say. Okay. You will have to know that one. I have 5 minutes left, so I'm trying to hit the. I'm trying to get to the more important ones here. Okay, pancreas. You have to know about the pancreas. What does the pancreas make? Insulin. What else? Glucagon. Exactly. More specifically, we have islets of Langerhans, alpha and beta cells. Beta cells are going to make your insulin. So we have specialized cells in the pancreas for doing this. We highlighted glucagon. We talked about it a lot, didn't we? Yes, we did. What do I want you to know about this? What do I want you not to know about it? Okay? Do I want you to know every individual thing that's happening here? No. I just want you to see that what? We have a hormone on the outside. It stimulates, it's G protein, adrenaline cyclase. It stimulates a lot of things going on. We ultimately start a protein kinase A. Point, hormone on the outside, lots of internal changes leading to an internal change on the inside. But what I do want you to know about glucagon, and the reverse of this is for insulin, glucagon does what to blood sugar levels? Increases or decreases? Increases. And how can we increase something? Well, let me ask you this. How does the cell store glucose? As glycogen. Roger that. Stores it as glycogen. So what can we do? I ask you this. What can we do if we want to increase blood sugar levels? We store it as glycogen. What two things can we do with glycogen to increase blood sugar levels? We can say, start breaking it up, and then we can say, stop making it. And by the action of protein kinase A, that whole type of GMP pathway turned on PKA, protein kinase A, it led to the turning on of glycogen breakdown. It also led to the turning off of glycogen synthesis. See, the red means off, green means on. Glycogen synthase turned off. Ultimately, glycogen phosphatase turned on. So we stop making it, we start breaking it down, our blood sugar levels go up. The reverse is true for insulin. We didn't do the whole insulin pathway, but it's the same thing, but kind of in reverse. Insulin, what are we going to do? We're going to turn on the synthetase, and we're going to turn off the breakdown of glycogen. Okay, I'm not gonna say draw this pathway out. I will not say that. But I do want you to know the two strategies that lead to it. I want you to know a series of intercellular pathways leads to the production of protein kinase A. But I'm not going to say draw the G protein moving after this, then turning on cyclic AMP, then turning on PKA and PDE and all these other things. We're not going to get that level of detail. But I do want you to see what is going on. Yeah, me too. Okay, pineal gland, melatonin. As for sleepiness and day and night cycles,